Is it good? Yeah. Awesome. Ready? Cool. Good morning, everyone. I would like to thank the organizer for this opportunity to come and speak here. Following up to the previous talk, I would like to expand on what Dong and Zhao just presented and give an example of our effort on creating a mid-density panel of our favorite fruit, blueberry. So as we know, blueberry is a diploid. The different species in vaccinium ranges from diploid to hexaploid, and it is one of the very important food crop because of its nutrition, known as superfruit, because of high level of vitamins, mineral, and antioxidants. Additionally, we consume it in different ways, from fresh fruit to frozen fruits, as well as juice and jam. And because of its wide range of market, it is economically important crop in US. Grown in different states in US, it is highly important. Only in 2022, the production was 622 million pounds was harvested from cultivated blueberries, whereas 77.6 million pounds of blueberries were collected from the wild. And this leads to a total of around $1 billion US, which is a very huge amount and contributing to the economy of US. But there are challenges for breeding this important fruit crop and our favorite fruit crop. One of them is long breeding cycle. It takes around 9 to 20 years to release a cultivar from its cross. One of the solution is using marker-assisted selection using markers and genotyping tools. But developing those genotyping tools, there's another challenge because of its genomic complexity. It's a polyploid nature, has a lot of genomic complexity. But if we can develop different tools, a different application, and adopt them geared toward these polyploidy nature, it would really help us. However, for that, there's some cost associated with it, as well as we need some bioinformatic knowledge, genomics knowledge to deal with these challenges. So breeding insight, as Dongyang mentioned before, we are putting a lot of effort to solve these issues and challenges for the breeder. So one of the effort that I will talk today is a mid-density genotyping platform or a genotyping panel that we have developed for blueberry, which is very quick, rapid, as well as it's cost-effective and very efficient. That's what we are looking for, as well as very flexible in design. If any breeder wants to change anything, it's very flexible and can be modified accordingly. So about the creation of this 3K dark tag panel, we first used selected a diversity panel of blueberries, 31 cultivated blueberries. Among them, 12 of them were northern high bush berries, 10 southern high bush berries, 2 hybrid of northern and southern high bush berries, 5 rapidites, and 1 cross between rapidite and southern high bush berries. So we performed a whole genome sequencing for all these diversity panel. Sorry. And we use these tools to process the raw reads. So Trimatic to filter them out, and then Boamim to align it to the haploid set genome of the blueberry, and DNA-seq pipeline by Sentient to perform the variant calling. After that, we collaborate with DART to finalize this 3K dark tag panel. A little bit more information about the panel, how the filtration went out. I know it's a lot of information, but I can talk about it later on as well. But overall, we had different steps where we thinned out as well as figured out based on QC from 600K, 10K to the final 3K that we have selected for the panel. And this is an overall summary of the number of markers distributed throughout the genome. Overall, we have in average 250 per chromosome, as you can see here, making it a grand total of 3,000. So now we have created the panel, we have it already. So the next thing we have to do is validate it to see how good it is, whether it works properly or not. So for that, we use couple of approaches to validate the dark tag panel. One of the approaches using another diversity panel to test the effectiveness, efficiency of the panel. So for that, we selected 375 samples that included a diverse set of cultivated blueberries, F1 population from a cross of Draper and Jewel. We also have wild vaccinium species and other interspecific hybrids. We also included 5 cultivated cranberries. So the first thing we looked at was whether the marker panel worked well or not, how much of the missing marker loci we found. So this is a summary box plot. As you can see from the box plot, the y-axis represents different groups of blueberry that was used, and x-axis represents the fraction of the missing data. As expected, we have high missing data for cranberry, which is a distant species, but for others, we had relatively less number of missing marker loci or missing fraction. We also studied the population structure. How good was the genotype cause to study the population structure? Because that's something a breeder or anyone working with different diverse germplasm wants to study. So we see a very consistent and distinct pattern based on the group that we have selected them for. For example, the cluster of dark orange or brown, light brown color that you can see on the left is a separate biparental F1 population, which is what we expected. Whereas on the right, there's that is the whole diversity panel, a different diversity panel. So this was one way to validate our panel. The second step that we used was try to see if we can construct a linkage map from the data we generated or not. So we used a biparental mapping population, an F1 population from Draper and Jewel, a northern high bushberry crossed with a southern high bushberry. And we also included 5 parental each. This is a general outline, a very general outline of how we constructed the genetic map. We had, so something I want to highlight is the missing parent imputation. So Draper and Jewel was developed after its release, has been used over the years, and both cultivars cloned of both cultivars have been used and distributed by different breeding programs throughout US. So what we assume was for the population that we are using for mapping, the parent may not be the exact same clone. So we were not sure, therefore we sampled 5 of each, Draper and Jewel, to make sure we can predict or identify the right parent. We identified the right Draper parent based on identity by descent method, whereas we were not sure about the Jewel parent. So for that, we had this missing parent imputation step, which I will explain further in my next slide. After that, it's all about quality control, genome-based mapping. Just to summarize, for genotype doses calling, we use UpDoc. Parent imputation was done by R and Excel, whereas other steps was done by MapPoly to construct the genetic linkage map and finalize the map. So talking more about imputation, the approach that we took, we imputed the Jewel parent mainly based on segregation pattern that was identified in F1 population and the parental genotype of Draper. So we did this whole calculation of all the expected segregation ratios that can come up crossing either having a parental doses from parental 1 and the other doses from the parental 2. We discarded the non-segregating markers from the whole population and focused only on the simplex segregation that I presented here because it's easy to predict those, obviously. So with that, we were able to impute 913 markers throughout the genome. And with that 913 marker, we selected the best Jewel parent among the 5 that we sequenced for our linkage map construction. So this is the linkage map that we got on the genetic map that you see here. We got a fairly good genetic map considering the imputation and best parent uses. Compared to previous genetic maps, it is fairly consistent, except a couple of chromosomes we found a greater genetic distances, but overall we would call it a very consistent genetic map. On the right-hand side, you see a comparison between genetic and genomic position, which is also fairly good. As you can see, I would like to highlight some of the features where we were able to detect some centromeric region. The one I highlighted in red box in chromosome 2 and chromosome 3, as you can see. So overall, with these results, we were very confident to say that the panel, that mid-density panel that we developed was good enough and efficient for these kind of works. So this is a summary of the genetic map. We were able to construct the map that I showed you using 725 markers. As you can see, some of the markers have big distances, but again, considering the imputation and the best parent uses, this is the best we got from our analysis. Besides this, we also looked at haplotype reconstruction, whether we would be able to perform a good haplotype reconstruction of the progeny or not. Again, it was done in MapPoly, where it has a function. The package has a function where we can do that. This is mainly done to see whether if any breeder or anyone trying to find map a certain gene can identify a F1 or a certain population that has a recombination breakpoint as their targeted region. So this is a example of two F1 progeny, the haplotype reconstruction. As you can see here, it's a complicated graph. I would like to just summarize it, highlighting on one. So the one box I highlighted is one chromosome. Different color is different doses or parental homologue explained in the legend, as you can see on the right. And the x-axis is the map position, whereas the y-axis is categorized into four sections, but it's overall probability of the doses calls that you can see. So anywhere you can see a jump of different color, that's a recombination breakpoint where some breeder or some molecular biologist trying to clone the gene would be interested if there is a QTL nearby. So yeah, with that, I would like to conclude my talk. So we were able to develop a 3K DocTech panel, which is now publicly available and accessible to order through DART. We are currently working on the manuscript. It's under preparation. So again, our main goal was to create a very cost-effective, although there are other panels, high-density panels available in the market, but we were targeting a cost-effective and rapid genotyping platform for breeders, both private and public breeders. And again, as the previous speaker mentioned, our another goal was to create an open access system where a certain allele that was identified in one group can easily be compared with the other and see if something is unique or a common allele throughout different breeding programs and breeding projects. With that, I would like to acknowledge USTA, Cornell University, our team, which have been working collaboratively on this work, and NC State, also our collaborator in this project, and Tools for Polyploidy. Again, we are a very direct benefactor from all the tools, all the programs that have been developed from this workshop, so and funding organizations. With that, I would like to thank you, and if you have any questions, I'll be happy to take it.
Any questions?
Yeah.
Surprised to see, it looked like you showed there was 20% missing data for a lot of your cultivating material too. Is that excellent? Can you elaborate on that? I have essentially zero missing data on potato garden. That you have a minimum count, or what's going on there?
Repeat the question.
Yeah. So the question was, I can go back to the slide. So the question was why there's around average 20% of the missing data in the DocTech panel. So I think Dongyang can explain it more. She would help me, but I'll try to address it first. So basically, the diversity panel that we used and the one that we have been testing, a variation in that, as well as the efficiency of certain loci, it does affect that during the sequencing and calling. So 20% in this case, I think it's good enough because of the range that we've got, because in the biparental population, it's very less, whereas some ranges are high. So I would say mainly because of the diversity or differences or distance that we have between the created panel and the testing panel, but Dongyang can explain it more precisely.
I think that's exactly.
Mhm.
Because we use like when we selected the snakes that we use 0.25 of minor allele frequency, then they are infected because we have like a diploid, tetraploid, and hexaploid species in the initial that we are now holding up skin sequencing at diversity panel. So
I mean, because 20% can mean different things. It sounds like you're saying it's 20% of the markers basically are not informative, and you get no reads for those. It's not that on any given marker you have 20% of the samples from the data, like you can see in the rats.
20% of the markers, 20% of the 3D markers.
Okay.
But they are not
that works. The markers, the 80% of the markers that do work, you have essentially it makes.
Yes, yeah. And also the 20% is variable across different groups that we have tested. So it's not the same 20% for all of them.
Yep.
Thank you so much for the question.